Hello, everyone. Welcome to the New Jersey Mycological Association's Taxonomy Tuesday for June 30th, 2026. We take turns sharing from our devices mushrooms that we have seen, and we help each other with identifications, and we try to learn from each other and learn from the mushrooms. So with that, I'm going to ask if anybody here has something they would like to share.
I'll share some.
Hopefully, folks have started to find some things, particularly north and west. It's been a little damper. So, anybody have anything they'd like to share from their device?
I think someone said they have stuff to share. I do too, but I think I heard someone say,
Yeah, that was me. This is Tristan.
yeah,
Hey, excellent, Tristan.
Yeah, I have some stuff too.
Okay, super. All right. So, Tristan, are you ready to go?
Yeah, I think so.
All right. I gotta close the door. Somebody's trying to call here.
You guys can see my screen?
Yes.
hey,
Unfortunately, I didn't get a lot of photos because I actually planned to go back the next day and take another photo, but it wasn't there. I don't know if somebody grabbed it or what happened, but and I tried to figure out what it was. I mean, so
Not a Campinopsis.
no, it's not.
Nope.
it's the Amanita.
That's what I
Yeah,
thought too. I couldn't
Amanita.
remember.
It's an Amanita from section Vaginata. Vaginatae.
That's funny. I not said that was so pronounced.
No,
Yeah,
I mean,
AI sucks.
Yeah,
that
that's
was that was
yeah,
my guess. That was my guess. I tried to look it up and I tried to figure out what it was, and that was like because I don't know. You see this page, I guess.
Yeah,
The,
oh,
this one is not an Amanita.
right. This is this is what I went off of. I mean, it kind of looks similar with
yeah,
the striations going down the cap there, and so that was my suggestion of the Coprinopsis. But
okay.
I mean,
You
I
could
originally I thought it
continue.
was Amanita with the sac on the bottom, but I couldn't figure out which one it was.
Correct. Yeah. So it's probably Amanita rooseveltensis. That would be my guess.
Okay. And then here's this was a different one, not that one that I showed there, but this was next to it. Somebody had, I guess, had broken it off.
Probably the same thing.
Yeah.
Nice.
Yeah, so the key features of Amanitas from section Vaginata include a totally elongating bulbous stipe. So there's no bulb, there's no thickening at the bottom. A saccate vulva, sometimes could be friable and not well structured. No partial veil, no ring, that is, on the stipe. And the caps are often not often always striated. You can see striations on the edges of the cap that can be quite long or maybe not as long, but they will always be present even at the early stages of development. And the typical colors are brown, gray. Sometimes you have yellow brown or more brown section vaginata, but the colors are fairly modest. They're not bright as some of the colors in the other sections of the genus, like Validae and section Amanita. But they're fast growing and they don't last very long, and typically they are fairly small. But sometimes they can get pretty big too.
I went back to this was like maybe like 7 p.m. I took it, and then I went back the next day, next early afternoon, and it wasn't there anymore.
It wasn't there because it could have been eaten by squirrels or deer or some other critter, but they grow pretty fast and they're gone within a couple of days typically.
Is there usually no warts on the cap?
It varies. Sometimes there are no words. Sometimes there's a big remnant of a veil, and sometimes there are multiple patches of veil on the cap. It's just quite variable. It depends on what species it is and what the conditions are, and so forth.
Oh, okay.
But if I recall correctly, this is one of the younger sections of the genus, and they're not as robust or long-lasting as some of the ancient sections like Lepidella, for instance, which are clunky and big and white. Colors developed much later in, along the evolutionary tree of this genus. So yeah, that's the kind of a typical look, and the cap, of course, expands completely in age and becomes totally planar or flat. But it's bullet-shaped like that when it's young, which is typical of most, if not all of the species in section Vaginatae. Dave probably has additional things to say, maybe something I missed.
I'll add something about
not much. I would say that one difference you see in Cortinarius scenarios is that the striations go all the way. They encompass almost the entire radius of the cap. The Amanitas, typically, you're not going to see striations much more than 50% of the
Correct.
radius.
Yes.
Yeah.
Good point.
And this does look like Amanita rooseveltensis to me, which favors grassy open areas. It's a pretty early fruiter. I get it on my property, but I haven't seen it yet this year. Usually I see it by now. And that one has a rather persistent membranous white basal vulva, as is seen in the photo that we're just looking at. And also sometimes the stipe has flocose deposits on it, as is seen in the photos. By contrast, say Amanita fulva, which is also a species of section Vaginatae, usually does not have much like scales or anything on the stipe.
Yeah, it's often the case that in the upper portion of the stipe, the upper half, the stipe in the rooseveltensis is very powdery. You can touch it and transfer that powder onto your fingers. That loose flocose pieces of it's probably limbus internus of the vulva. That's what I think it is. Internal limb of the universal veil. Which is present in all Amanitas, obviously, but it's presented differently in different species and different sections. Yeah, rooseveltensis is probably one of the earliest sections Amanita, Amanitas from section Vaginata to appear on the scene. And
that one and fulva.
Yeah, we collected fulva at Stokes just on Sunday. Lots of them.
I've already seen maybe 5 or 6 fulva over the past, say, 5 days.
Mm-hmm. Yeah,
just for
fulva is probably one of the easier Amanitas from section Vaginatae to identify owing to its very specific and stable colors, and as well as brownish orange staining on the vulva on the bottom of the stipe.
here's an example, I believe, is fulva, just for comparisons.
Yeah, it's darker on the disc. It usually has a little umbo. As you can see, the stipe is pretty bare on this one.
Mm-hmm.
It's got the basal vulva. Yeah. Striate cap margin. So Steve Russell, who does a lot of sequencing, says that different from a lot of the other species in section Vaginatae that vary in color, and sometimes there are ones that are the same color, but they turn out to be different phylogenetic species, different genetics. He says almost all of the things that look like fulva that he gets to sequence turn out to be fulva. And this is also an interesting species because it's in the minority of mycorrhizal species that occur naturally, not just because they got moved on trees, in both Europe and North America.
Yeah, I think it was Rod who determined eventually that what we have in North America is the bona fide European fulva, not the Ameri fulva, as you called it before.
Yeah, I'm guessing that he had made somewhat of an assumption to begin with, that thing, because everything was turning out to be different when it was found in North America. And I think once he started sequencing, that's when he switched, when he changed his determination on this.
Yeah, I think there's probably some distinctions between the ITS sequences or nuclear ribosomal, genes in the American fulva versus European fulva. But when you build trees from those genes, it's just going to be sort of one big clade. And there's still a possibility that what we have in North America is a vicariant species, so it should be named differently from the European fulva, but this is still kind of arguable. This is kind of work in progress. So for now, we say that fulva is present in North America, which is unusual, for amanitas and other fungi. Typically the mycota of Eurasia and North America are very, very different, and there's not much overlap, with a few exceptions. For Amanita and other genera, that is.
Yeah, for mostly for mycorrhizal species, they
Right.
seem to have diverged more
Mhm.
than saprobes, which is understandable because saprobes probably get moved around a lot easier.
Yeah, but even here you can get, geographic isolation. I think what's the term for that? Sympatry or allopatry? I forget. I have to check those terms. But yeah, typically that's what happens. You have some kind of a geographical divide, and eventually you're going to get speciation, isolation of gene flow, and all of a sudden you're in a different branch. You're diverging away from your parent species. So that's always the possibility, and that's why we need to sequence not just like one or two genetic markers, but whole genomes. Because some genes mutate at a different rate.
All right. Tristan, do you have any others you want to share?
No, that was it.
That was good. All right.
What was the name? I mean, that was so started with an R. Amanita.
Rooseveltensis. Rooseveltensis is named after the town in New Jersey where Rod happens to live. Roosevelt Township.
If you use a microscope, you can verify to a pretty good level of confidence that your gray Amanita section vaginatae is Amanita rooseveltensis by looking at the spores at 400 times magnification or more. 400 times is enough for this. They would be ellipsoid or broadly ellipsoid and not globose or subglobose, meaning round, as is the case with almost all the other species in section vaginatae.
All right,
But there are exceptions. For instance, Amanita berry color can have both globose, subglobose, and ellipsoid spores.
Yeah, do you find that in New Jersey, that species?
Oh, absolutely, and it's a chameleon. It can be almost white or it can be dark brown.
Oh, you get that in the Pine Barrens?
Absolutely.
Yeah, because I've never seen it here.
Yeah, it's been proven genetically. I've sequenced, ITS and TEF1 genes, and it's the same thing, and looks like it's a good match to the original species from Canada. I think it's
that one would not look much like rooseveltensis, though.
No.
So you wouldn't really be apt to confuse those two.
No,
I
it
think there
has.
are a couple other gray-capped ones that have ellipsoid spores, but they're sort of regionally segregated. Like I think maybe there's one from Louisiana, I believe, Baton Rougensis, maybe. I think that one might have ellipsoid spores.
Yeah, there's a number. There are a number of gray section Vaginatae, and it's a difficult section, and there are many species. And you have to do a lot of sequencing and hope that somehow you can build up species concepts after you sequence enough of these things.
Yeah, we're still finding genetically different species in section Vaginatae.
Yeah, we're still at the early stage of discovery where we're not done yet with anything.
All right. Let's move on and discover some more mushrooms to look at here tonight.
Absolutely.
All right. So Tristan, that was the only one you wanted to share.
Yeah.
So okay. Then let's move on. Who else? If you've got some, you can put your name in the chat, or we're a small crowd tonight. Just speak up, please.
Hunter wants to go first.
Oh.
Ladies first.
I was gonna say Igor should go now, but I can go. We'll go. Here we go.
All right.
All right. Can you see Mushroom Observer?
Yes, you're
Okay.
scrolling through your thumbnails.
Yeah. Okay. Cool. Look at this one. So, this presentation was a little bit interesting. I could tell I could tell it was a Pleuteus, but it was they were kind of growing cesspitose in this cluster like this. But that
Petasatus.
pardon?
Pleuteus petasatus.
Yeah,
Oh.
petasatus does this. It grows in clusters, and it grows in the open often. Not always, but often.
Pedasadus.
I think there's some T's where you said B's.
Yeah, not quite. I'm going to put it in the chat.
Yeah.
Thank you. Okay, excellent. Yeah. So yeah, so I knew it was a Pluteus, but I did do spore prints, and well, you could see the spore color here on the cap, and also even on the gill edge of the face-off one. So it's that pinkish orange Pluteus color. And then the horned cystidia, right? Cystidia, right?
Yeah, those are pleurocystidia there that have more than one horn. One thing you can look for if you think you have petasatus, you can look for what they call intermediate cystidia. They're sort of like pleurocystidia and sort of like chylcystidia. But what distinguishes them is they have they don't have multiple horns. The intermediate cystidia have just come to a single point.
Oh, okay.
So this one that's on the bottom here, that one looks like it's verging on. That one's got two horns. They're pretty small though. Yeah, but you can look around for those kind of cystidia.
Okay. Well, this is great to put a name on this one. Thank you. And so what else do I have here? Foliota polychroa. And this I recognized by sight. It's one of the easier foliotas to recognize. Its color varies a lot, but it's colorful, and it grows on wood. And I did the spores, and they look like this. And that was that. So if you haven't been here, Lila, I've just been trying to get down a rhythm and process to my microscopy and just do it, even if, just do it to get the rhythm going. So that's what I've been doing, and I'm continuing to do that.
Great.
Yeah. Yeah. Although, yeah, I'm realizing that spores. Well, yeah, I'm working on it. So, and then this I assumed also was Mycena haematopus or topus, but the bleeding Mycena, but it didn't bleed, which is why I just had left it at Mycena. I could get no blood from it in this stage, or when I actually picked it today and got the spores. The spores are very large, and they're this isn't a great picture, but they're heavily ornamented. They're really neat, and this picture does not do it justice at all. But it looks like they have a little, maybe a piculus, and then this one, I don't know what's coming out of that one, but it has a little something on it.
Looks like it might be an air bubble stuck to it.
Oh, okay. Yeah, that could be.
Did you squeeze one of the fruit bodies? Sometimes you need to do that to see the red juice.
Well, I tore them, and yeah, I did try very hard to get the blood out, and it just didn't happen. And it wasn't dry at all this day, you can tell. So yeah, I don't know, but
There are some traits here that point very strongly toward this species. I believe the tooth margin is a trait of haematopus. I'm pretty sure. I would want to look that up, but I think it is.
Dave, I agree with that. The triangular, the triangle shaped margin.
Yes. Okay. Well,
growth in clusters on wood. Yep. And pretty good sized mushrooms. There's another bleeding Mycena, and who knows, there might be a few other species hiding, in the two names that are typically used. I think haematopus is probably pretty well settled, but there's at least one smaller one that grows usually in moss, and it grows singularly, or you might see two or three. Usually there's only one. Mycena sanguinulenta, and that's a small bleeding Mycena.
Yeah, and Dave, I think you're to your point. Even if you find two or three, that one would never be in a cluster like this.
Never saw it. Nope, in a cluster. Always growing singularly.
And Hunter, did you? So if you have I assume you carry a knife.
Yeah.
I always get these things. The best way for them to bleed is to cut the cap. So make like a little slice in the top of maybe the biggest one, and that always seems to bleed pretty good.
Okay. Rather than tear it, yeah, because I tore it, and that didn't work. So, but I'll try the knife next time.
Yeah, if that doesn't work, just take a cap and or take a stalk separately and squish them between your fingers and see if you see a little red juice on your finger.
I did, and I mean, yeah. If there was any, it was like really scant. I was So yeah, I don't know why, but yeah, I totally agree with the ID, and they're very pretty. So yeah.
One more thing, actually, Hunter, in support of the ID here, I have found them when they appear fresh, and even smashing them between your finger, you can see a little bit of like red juice coming out, but then once you release that, it kind of sucks back into it. So yeah, I think sometimes it's not all that obvious.
Yeah. Yeah, okay. Well, yeah, it's
And then other times it's like, oh my gosh, this thing is, I got red juice all over me.
yeah. Well, that's what I was hoping, but it didn't work out. But yeah, thank you. And then I found this Cortinarius, um, and I think it's distans. It was growing in grass. There were multiple ones.
There any oaks nearby?
Yes, large oaks right there.
Yeah. So this name might very well be traditionally applied to more than one genetic species. There's a section that includes distans that may or may not be holding up to phylogenetics. I'm not sure. But there might be more than one phylogenetic species that looks like this, and they're probably related genetically, but not exactly the same. But the things I look for are three things. It's an early fruiter, right? So it's one of the first courts you see. It's usually with oak. It has the widely spaced gills, like these do, and it usually forms a ring on the stalk as opposed to just a little webby sort of ring zone. And you can see here there's an actual ring there. I mean, it's not a real big ring like you see on some of the Amanitas, but it's a well formed ring. Also, the cap tends to be a little sort of finely grainy, scaly toward the margin on this one.
Yes,
So
yeah.
so this might so this one might be a little. I think I've had things like this sequenced, and I think at least once um, I came back with a different name than distans, um, but that's a good name to go with. In the absence of more advanced kind of analysis. Also, the spores tend to be they have a small Q value, so the width is those are a little that Q looks a little larger than I would have guessed. Now maybe I'm just forgetting. Um, but certainly the mushroom looks like Cortinarius distans.
Okay. Thank you. Um, yeah, and then if you want to just quickly, so I found this Agaricus in grass here. And um I didn't I did stain yellow with KOH, but I didn't do the rub. You can see it has this huge cogwheel veil, but I didn't really remove the dirt to examine the base better. But then also, and that was growing in grass. And then I also found these growing in conifer, and these also stained yellow with KOH. They also stained yellow when I rubbed it on the gill edge, on the cap edge, and also on the base of the stipe. And I also found some in oak woodland. And these also stained yellow in KOH. The stipe base stained yellow when I rubbed it, but I think this one was too mature, so when I rubbed the edge, it came off. Instead of staining, the cuticle just kind of rubbed off. But I looked back at the NJM newsletter that has the article I think Sue wrote about Agaricus, and it has a chart to put them in sections. And then it also refers to, I think, the Kerrigan key. And I started really just to look at those charts. And I mean, one of the things, and so why I bothered to, like, even after I saw that first Agaricus, why I bothered to make observations for the second two was because the habitat is definitely something to consider when IDing, but it's certainly just one thing, not the only thing. And then apparently species can, sort of, it's not a rule. And just because you find it in grass doesn't mean you won't find it in a woodland as well. But they're in this chart in the yellow staining Agaricus, they kind of divided them up as a woodland species, grass species, and conifer. So that's what I did. And then I didn't smell them, which I guess is not, as you definitely want to smell them. So they might have an almond or sweet scent, but otherwise they might also have like a was it creatone scent? A bad, pardon?
Phenolic.
Phenolic. Okay. Yeah, in the chart they say like creatone.
Yeah, maybe they say that too. Now, sometimes the ones that smell like phenol, and most of those are toxic. They won't kill you, but apparently you can get pretty sick if you eat them. Some of them, sometimes they don't smell. Sometimes you can't detect that odor.
And
the yellow stain on the bottom of the stalk is usually, you'll see that on these ones that are like related to Placomyces. Oh, there's some yellow there.
Yeah.
Yeah.
I think the second species that was growing with conifers could be approximans. Agaricus approximans. Yeah, this guy.
Ah.
Can you put that in the chat, please?
Yeah.
Okay.
Yeah, Agaricus ID is yeah, it's quite challenging.
Yes,
usually we rely on sequencing for that.
Yeah, except a lot of people will eat the meadow mushrooms and the horse ones. I mean, there's people who regularly eat agaricus mushrooms that don't know any of this other stuff. I don't think.
Yeah, that's the ones that people eat are basically fall into the two general categories: the horse mushrooms, which are a section arvenses. They're large. Sometimes they develop a little yellowish or bronze kind of stain, especially on the cap surface, but they're large. The gills, if they have a pink stage at all, it's very short lived. They go from like beige, grayish beige, right to brown, but they're very large and they grow usually in the open, and a lot of times in the open with spruce trees or other conifers. But they're really large and they smell nice. They smell almondy or and the other category that people eat are the meadow mushrooms, which are fairly small. The partial veil typically, and the partial veil on the horse mushrooms, it's a cogwheel partial veil, and it forms a fairly persistent ring. The horse mushrooms, the partial veil often is not all that prominent. You might get a little ragged ring on the stalk, but they're kind of small and they have very vividly pink gills when they're young. And they don't have much of an odor, but the odor they do have is rather pleasant. And there's usually they grow in root groups on lawns or in fields.
Okay, and so none of what I found was that, even the one in grass.
I don't think that any of these three that you have fall into either of those two categories.
Okay, and are the red staining all uh inedible? Because then there's a whole group of Agaricus that stain red, which none of these are. But are those all inedible or
I've never eaten any of those.
okay?
The only agaricus that I've eaten are um the horse mushrooms, the meadow mushrooms, what I have called abruptibulbus, which is mainly a woodland species, but it has a very pleasant odor. And
The little hook foot does it?
yeah, it's got an abrupt bulb at the bottom of the stalk.
Okay.
And I think it might also have like a double veil,
Okay.
a partial veil. Um, but I don't know. The more I've learned about agaricus, the less I'm apt to experiment with species that I don't know.
Yeah.
Because a lot of them are said to be toxic, and it's such a big genus with so many different species.
Right. Well, I'm just kind of trying to get to know them better, and it was fun to do this rubbing with my hand, my thumb, and get this yellowing reaction with, and not without even using chemicals, but then also the yellow flash with the KOH was cool too. So yeah, it's just, again, just like trying to get the methods down. So thanks. That's it for me tonight.
Yeah, this Agaricus could be one of those three species that we have that sort of fit into the campestris group. But again,
Is that the meadow mushroom group?
yeah, well, we don't have campestris per se, which is, I think,
You
European.
you think this might be? Look at the partial veil though; it's really big and persistent.
Yeah, I know, but there will be other species that grow in the grass, but these always grow in the grass. The three species,
Uh-huh.
Brandelii, I forget the other ones. I just cannot recover the names at this time. I know where to find them, but I just cannot come up with the epithets at this time.
Yeah, I mean, maybe this is related to what we call the meadow mushrooms, but I would not think, if I found this, I would not think of it as a meadow mushroom because of the really persistent ring on the stalk. It's very
Yeah, maybe.
large.
I mean, it could be something else because there'll be other species that just like to grow on grassy,
Oh
meadow.
yeah, I've found plecomyces in grass.
Right,
Um,
but this doesn't look like Plecomyces to me, though.
you should see a little more ornamentation on the cap and more yellow in the base of the stalk
Yeah.
with plecomyces.
What's a common name for Blaccomyces? Is there one that you know of?
Hmm. I don't know.
Yeah, I'm not big on common names for mushrooms in general. I don't like common names.
Okay, so
Yeah.
nobody eats Blaccomyces, or do they?
Well, maybe some people do, but they're considered to be toxic.
Oh, okay. Okay.
Yeah, the greatest danger is that you have no idea what you're eating. You assume something, and even though if you don't get poisoned, you still have no idea what species exactly you're consuming, and
Yeah,
therein lies the danger. And you always want to know what you eat.
absolutely.
Um, I mean, some people are comfortable eating things to a genus. I prefer to know the species.
Yes,
That's
me
just
too.
me.
No, no, I agree 100%. No, I'm just trying to get to know them, and that doesn't mean I'm gonna, I'm even eager to eat them. It's just wanting to know it better. So,
Yeah,
thanks.
the one last thing I want to say about this particular agaricus here, that the properties of the cap, the colors, are not consistent with the campestris group.
Why?
But who knows? This thing was exposed to the sun, and variations happen, and you've got to really build up your concepts carefully with bona fide data, including DNA, to draw any reasonable conclusions.
Okay.
Or you could buy the giant Kerrigan book,
Yeah,
and
yeah.
it also doubles as self-defense because you could beat somebody to death with it.
Yeah, it makes a nice doorstep. I mean, I still have my copy, still wrapped in original plastic. I never opened that thing. I'm keeping it. Maybe I can resell it for twice the amount I purchased it for
You probably could.
in 10 years or something. Yeah,
That's funny.
Beautiful.
it's already out of print, so things are looking up.
There you go. All right. Well, I'm gonna stop my share.
Thank you, Hunter.
Thank
Thanks,
you.
everyone, for the discussion there. Okay, I think Brandon had some, Igor had some. Actually, Brandon emailed, so before we go to the email, Igor, do you want to go next?
Yeah, sure. Let's see. No, I don't want to share that. This one. All right, we're going to start with this fine example. So we had a foray at Stoke State Forest, and this is the northwest section of the state, where you have more sort of boreal woods with mostly hemlock, some other conifers, oak, birch, maple, but it's quite a different habitat from other parts of the state, especially the Pine Barrens or Middle New Jersey, which is typically all broadleaf. So finally, I can say 100% that we have Neoboletus subvelutipes in New Jersey. After so many years of looking for this critter and not being able to sequence anything other than its next of kin, which is a different species, you can say without a doubt, just by looking at this specimen, that this is a bona fide Neoboletus subvelutipes. And this is the first red-pored bolete that was described from North America by Charles Peck. And for a while, it was a very controversial species till the type collection was finally sequenced a couple of years ago. So now we have both the genetic data from the type, and we have genetic data from modern collections, and hence we have a nice species concept for this taxon. So Neoboletus subvelutipes likes to grow with conifers. It's not a hardwood-associated species. And that's very important to remember, because that's going to basically steer you toward the right identification, because there's a lookalike of subvelutipes, as I mentioned. It's called, unfortunately, it's called subliridalis. That's going to be the proper name for it when it publishes as a comb nov, a novel combination, because it still sits in genus Boletus. But the key features of subvelutipes, other than the ecological trait that I described, conifers, hemlock and Norway spruce, the colors of the cap, which are yellow-brown, maybe tawny, although it varies. This type is yellow with flushes of red, which are arranged in this kind of a network of streaky, reds, like tongues of flames that are rising. Okay, it's not dotted; it's more like streaked. It's not going to be reticulate, although it could be reticulated at the apex. So you can see the reds, how they show up on the darker yellow background. And of course, it's going to blue all over, not only when you cut it, but, the cap is going to stay in blue, and the stipe is going to stay in blue when you touch it or rub it. And on the bottom of the stipe, you have the mycelium, which is yellow. You have all these hairs, preinosity, not preinosity, strigosity. That's going to be either dark yellow or dark red, and hence the name subvelutipes. It's velutinous. It's hairy, which is not a very descriptive term, because other Neoboleti have similar arrangements of mycelium on the lower stipe. So when you find a species a few times, you'll sort of recognize the overall gestalt. So it's going to be pretty intuitive. Again, the major thing is that it has to be conifers. If your woods are hardwoods, you will not find subvelutipes in those woods.
So this is what I showed a couple of weeks ago, last week in hemlock, right?
I think so.
Yeah, I think so. Okay.
We found other collections of subvelutipes. Other people brought it in, but this is like the first year where I can say, yes, this is subvelutipes, and we do have this in New Jersey. I mean, I suspected for a long time that we've got to have it in those woods because Dave finds it a lot in his neck of the woods, and the habitat, the ecology there is very similar to northern New Jersey. Hemlock woods with birch, oak, and other hardwoods mixing in, but predominantly hemlock woods.
Exciting. That's exciting.
Yeah.
How about the stature of this? Is that a typical size? Is that about how big they get?
Well, this is still young. I mean, this specimen was in pristine condition.
Yeah.
I think it's Svetlana's. She took it home to eat. I think I told her she would be okay with it because Dave ate subvelutipes and nothing happened to him. Right, Dave?
Yeah, only maybe 2 or 3 times. At least one of the times I parboiled it. I'm not sure if I did that each time I ate it. I may have just fried it up one time, a small amount, but I don't routinely eat these.
Yeah, I told Svetlana to parboil it or cook it very thoroughly.
Yeah.
And she should be okay.
I got an email from her yesterday, so.
Oh, she ate all of them?
I don't know. She didn't say anything about eating it, but just that she's still with us because she emailed me yesterday. So
Well, she's not going to be poisoned to death by any of these things, even if
no.
they are, mildly, quote unquote, toxic.
Yeah.
So that's that. Very iconic, easily recognized species with enough experience. And then we have, this thing, which is from a different genus, and that species was described recently in a paper by Colin Damnauer. This is Lanmaoa carbonilivor, and the name, the epithet basically translates as charcoal stain. And this is a name. It's in a positive, so it's a noun. So you have 2 nouns. So don't be confused by the ending of the word because it is a noun, not an adjective. So this species was first recognized as something unusual exactly from Stoke State Forest at Aquetank, this foray site back in 2017. And that's what I thought was Boletus sensibilis, because somehow over the many, many decades, since sensibilis was described by Peck in the 1800s, one of characters of that species was attributed to it, the curry smell. And interestingly, that smell is not in the description of sensibilis. So who knows what Boletus sensibilis is? It might as well be the species. But for the time being, this curry-smelling Lanmaoa is called Lanmaoa carbonilivor. The key characters, other than the odor, are the pink caps that fade away to yellow or yellow-brown from the disc outwards, not the other way around. And the pinks are fairly muted. They're not bright or rich pinks. They're fairly, I wouldn't say pale, but kind of mild or medium tinged pink. The stipes are also pale yellow, and they can be flushed with reds, but for the most part they are yellow. There's not much red in them typically. I'm trying to increase the size of these pictures, but they keep reverting to the original size. No reticulations, and this bolete stains blue I wouldn't say very, very heavily, but yeah, it will stain blue prominently when you touch it. And of course, when you slice it, which I didn't because I wanted to keep it intact for people to look at the foray. When you slice it, the flesh is going to be staining blue everywhere uniformly, not just patchy here and there. It's going to be a uniform blue color through and through, but not necessarily very dark, although it can be dark. So again, boreal distribution above the Mason-Dixon line, albeit this species was found in Virginia at high elevation, 3,200 feet, in the Shenandoah National Park. But again, boreal distribution, the curry odor, the pink caps fading to yellow-brown, pale yellow stipes, heavy bluing or relatively heavy bluing is a telltale, other telltale features of the species as opposed to the other Lanmaoa species. You can basically identify your Lanmaoas by key traits or by exclusion principle. You can eliminate other taxa from consideration because they lack certain properties which could be unique to particular species. We found the species at Meadowwoods last year, so it's possibly associated with both oaks and hemlock. That's my suspicion, but it's not proven. All right.
What's the pore layer? How deep are the tubes?
Oh, well, like all Lanmaoas, the tubes are going to be fairly shallow. And I think it's not just the depth or the length of the tubes, it's the ratio between the thickness of the cap in the middle, not where the stipe is, but in the middle of the radius, versus the depth of the tube. So 3-to-1 ratio is quite typical for Lanmaoa, 3-to-1 or greater, with 3 being the thickness of the cap.
Yeah.
So the cap is at least 3 times as thick as the tube layer.
Yeah.
Yeah, here's an example of the same species, which is huge. Before we found specimens that were fairly small, but this particular cap was 6 inches, and it's the same species because it was found in exactly the same spot where a collection from 2017 was sequenced. So they can be quite large, but not often.
The first one, it has 2 stems, right?
Yeah, it's a Siamese twin.
I found one like that too, I believe. I might put it in the chat. You might want to check it out later.
Yeah, this is another Lanmaoa carbonilivor that was found only a couple of yards away from that big one. And sorry about that. Just to show more of the morphology of a different specimen. The picture was taken with a flash, but you can see the same thing. The stipes are yellow, devoid of any vivid reds, and the caps are pale pink, fading to yellow, yellow-brown. This is the cap, kind of be, a mixture of colors. This is what happens to this thing. So again, boreal distribution, mixed woods with hemlock, curry odor, pink caps, yellow stipes. Not much red in the stipe. Bluing pretty heavily. Typically on the smaller side. Now this bolete, which I like a lot and I don't see often enough, is another associate of hemlock. And again, when you are in the hemlock woods, be on the lookout for the species because this is the only place where you're going to find this. This is Boletus miniato-ovaceus, described by Frost in the 1800s. And although we don't have a type sequence, we're pretty certain of the species concept at this point in time because Frost's description was pretty good compared to those of Peck. He was a little bit more detailed, and this bolete is distinct. These are young specimens, so the cap colors are very rich. But when they get older, they fade away, and they also develop olivaceous stains. The stipes are yellow, flushed with these streaks of red. The pores are yellow. It's going to stain blue, but not much. It's not a heavy bluer. But the flesh is going to stain blue too, but the flesh is pale. Dave has a lot of experience with Boletus miniato-ovaceus, and by the way, it is going to be a member of genus Boletus in the future.
Yeah, this one, the flesh in the cap is almost white, and there's often some slow staining, blue staining inside the cap. Not
Right.
much staining inside the stipe, which is also rather pale context, but a little bit more yellowish. Sometimes the pores will bruise fairly darkly. Notice how the stipe is pinched at the base.
Yep.
That's a trait of this species. And as Igor said, as these expand and go past maturity, the cap develops patches of olivaceous, like greenish color. Hence the species epithet.
Right. So the smaller specimens I found growing from the root of a hemlock. It was actually growing out of wood,
Oh, huh.
which is unusual. And when I smelled it, to me the odor was faint, but it smelled like medicine, maybe a little bit chlorini, but it smelled like some kind of a nondescript medicine, kind of a chemical odor. And later on, I just couldn't smell anything at all, so it was very faint. It was not a fiction of my imagination. I smelled it, more than once, but I'm not sure if that odor is going to be consistent, uh, for other collections. I need to smell boletes more often. This is something that I forget to do quite a lot, unfortunately.
Yeah, in some of the older literature, this is said to be toxic.
Yeah, you know what? Svetlana took these with her, and I think I did tell her that this thing has a mixed reputation, but she's going to try it. And I told her, don't eat
Oh,
a lot of it. Try maybe a small piece and see how you do, and definitely cook all your subvelutipes and all the cook all the bolete species separately, such that
yeah.
because if you mix them together in a blend and you, God forbid, get poisoned, you will never know which species caused the poisoning. But she took these homes, and I told her that it's not clear cut whether the species is entirely edible. Because to my knowledge, it's relative from the west coast, which goes under the name Boletus smithii, has been implicated in some gastrointestinal disturbances. All right, so let's get out of this one. Very nice bolete. I like it. It's very pretty to my eye. We got more of these red and yellow boletes, and this one I believe is from genus Hortiboletus. Unfortunately, the person who collected these things didn't do a good job extracting them from the ground, so they kind of came out mangled and slightly wasted. um, but I tentatively identify this as Hortiboletus campestris. Why? Well, it would be difficult to explain. It's not an easy genus. The most common species from that genus that we have in Eastern North America is Hortiboletus flavorubellus. And that species is quite variable. And there are probably some either subspecies or subpopulations that have a slightly different DNA, ITS, and you can build a very nice tree that's going to give you subclades. And I have a tree somewhere posted, and it works pretty well. But I believe this is not Flavorubellus. I think this is campestris. But I saved this, and I'm going to sequence it. It blues. The stipes are red, the caps are red, kind of a rhubarb red. Very nice color, very rich and pretty. And the pores are angular. They're more xerocomoid as opposed to boletoid. even though Hortiboletus is a member of subfamily Boletoideae, which typically has hymenophore presented as boletoid tubes, meaning that when you separate the tubes, they will cleanly separate from each other. They will peel away from each other because there is some gelatinous material between individual tube walls. And instead of breaking the tubes, you will be individually peeling them away from each other. But this genus, I think, has xerocomoid tubes, which are pores, tubes that don't have that gelatinous material between the adjoining tube walls, and the tubes are going to rupture instead of separating cleanly from each other. Small size, definitely Hortiboletus, name to be determined to a certainty with sequencing.
Igor,
So
the caps on these, are they cracked or no?
Hortiboletus caps are often cracked, yes. I've collected campestris in the past in upstate New York at the nymph, and it had a kind of a different look to it versus the gestalt of the Phylloporus group. And this particular collection, to me, looked more like campestris than Phylloporus. And by the way, the type collections have been sequenced. So now again, we have genetic data from the historic holotypes as well as modern collections, so we can build up species concepts confidently and maybe separate these species in the field. It's a very difficult genus, albeit it's not a very large genus. We only have a handful of species in North America, both in the East and in the West. Dave, what was your question?
Sometimes I find it to be a little bit confusing when the Hortiboletus, presumably Hortiboletus, when the fruit bodies are a little bit larger than usual, because I think they can then be confused with at least one species of Xerocomelus. There's one I know that sometimes has a red cap, or
Yes,
supposedly.
there is a cryptic Xerocomelus species in the East that was frequently identified as rubellus of Europe because it has this really purple red, vivid, dark color, very beautiful. And Colin Damnauer is going to describe it as a new species. I think the name that we gave that critter is Rubino diffractus. Rubino refers to the ruby red color, and diffractus means that it's cracked. I think on the Mo, it is named Boletus species Indiana 01 at this time. And it's a very distinct looking species. I think you cannot confuse it with, it looks a little bit more like Hortiboletus, but actually it's a Xerocomelus. It doesn't look like a typical Xerocomelus. It looks more like a Hortiboletus. But both Hortiboletus and Xerocomelus have cracked or diffract caps. What's the better word for that?
Oh my gosh, what is it?
What's the Latinese for
Aerolate?
Aerolate.
Yes.
Thank you, Dave. Yeah, right. So that's that. So that was again, a very unusual habitat, more of a boreal hemlock, woods with intermixed with some hardwoods. It's not a typical Hortiboletus flavo-rubellus habitat. That's why I suspected this might be something else, possibly campestris. I think I've exhausted my boletes. Here's another collection of subvelutipes, by the way.
Yeah, you just one more comment on Micah Quebec. The thing that I was thinking of, the Xerocomelus, they're calling Xerocomelus intermedius.
Oh yeah, that's another one, right?
Mhm.
Xerocomelus intermedius, I think, has been sequenced, the type, so we have an ITS sequence. I don't remember if I matched anything to it, but in my experience, Xerocomelus intermedius is also a boreal species. We probably don't have it in New Jersey. That would be my guess.
Okay, I think I found it here. But the thing that's confusing is, you know, it looks kind of like a large Hortiboletus. So, you know, can Hortiboletus get as big as some of these things I've seen? You know what? I'm going to look for an observation of mine and just put it in the chat. And
Yeah,
copy
yeah. I
it
mean,
later.
I need to go back and try to match, modern sequences to the historic, holotype sequences, and maybe there's going to be a collection or two on Mushroom Observer that can be confidently identified as Xericomelus intermedius. But it is indeed a member of Xericomelus. That has been confirmed. All right, I'm going to move away from boletes and maybe two more collections here. One Amanita and something else. I just want to talk about this very distinct Amanita that is very easy to ID in a field. This is Amanita solaniolens, and it's a member of section Validae because it has a unique combination of features for section Validae. Members of section Validae will always have a stipital bulb, even though it can be small and indistinct. They will always have a persistent partial veil, that is a ring, and their caps are not going to be striate, although there's maybe one or two exceptions. One exception is Amanita umbilicata, but I'm not going to talk about that Amanita because it's just outside of the scope of this discussion. But you can safely assume that the vast majority of section Validae do not have striate caps. So those three features alone should really eliminate everything else. And on the top of that, you can add the fact that a lot of members of section Validae have vivid colors. They're brightly colored, Flavaconia, Flavarubens. This one has a brown cap, which is not vivid, but again, it has some color in it.
Igor.
Sometimes these can be rather pale with maybe some pigment on the disc.
Yeah, but I'm just saying in general, Validae consists of taxa that are brightly colored.
Yeah, except for the white ones.
Yeah, except for the white ones like Canescens and what's the other one? Submaculata, that can be white when it's young.
Oh, yes. Yeah, it starts out white, right?
Yeah, I think those are probably early divergent species, and they're kind of lacking the bright colors of the typical section Validae.
Yeah,
but
by the way, I put a link in the chat, Igor, if you want to look at some point at my mushrooms my boletes that I found that I'm not sure if they're a large Hortiboletus or a Xericomelus.
Okay, I'll absolutely take a look at it once I'm done here with my spiel.
Right.
So Amanita solaniolens is always on the smaller side, and the ring is small but persistent, and it's tinged with yellow. The caps are brown, and they're going to have a kind of a sunburst pattern, with a darker disc. You see patches here of patches of the universal veil, the vulva, but it's not unusual for the caps to be completely bald, lacking any warts. The bulb is very distinct. It almost gives you an impression of being limbate or saccate, but these are just a thing limbs. And there's a smaller grouping within section Validae that has bulb that has species with the bulb that have that particular structure. These are your species of Lavengula, the Lavengula group. Brunescens has a somewhat similar bulb, and indeed they are genetically related. So do not be fooled by the unusual presentation of this bulb. It is not saccate. It's sort of pseudo-limbate, I would say. It's very squishy. It's very voluminous. It's rather distinct looking. And when you get familiar with the other members of that small subclade, you're going to get a distinct feel for the structure of this bulb. It's very unmistakable. And another feature of this Amanita, I think that it derives its name from is the odor. It smells like raw potatoes. And the genus, what's the genus for potato? Anybody knows that? For the plant of potato?
Is it
Sola
so-
A
something.
nightshade.
What was that, Chris?
It's a nightshade.
No, the Latin name.
I'm gonna guess it sounds like Mappy because I think that's where they got series Mappy from.
No, I think it's something Solani.
Yeah.
So this Amanita is named after a potato
Solanaceous.
because it smells like a raw potato.
Oh, okay. Oh, okay. Now I was gonna ask. I believe this species is also placed in a classification called series Mappy.
Yeah,
Does
series map
that
is
sits above Stirps lavandula.
yes, series is
Okay.
just above stirps. It's a rank that's above
Okay.
stirps. Yes.
Okay.
And
Yeah.
that's
So series
Stirps
mappa
citrina.
includes
Stirps.
lavandula, mappa, brunescens, and solaniolens.
And is that Stirps citrina, right?
Well,
That's
I
right.
have to
That's
check,
right.
but
It's Stirps citrina, not Stirps lavandula. Right. I'm wrong. It's
yeah,
Stirps
stirps
citrina.
citrina.
Thank you.
Citrina actually, people call it Amanita citrina. Citrina is not a legitimate epithet for any of the Amanitas from the small clade. Mappa is in Europe. Lavandula is a provisional name from North America. I don't think any of our um, series Mappae Amanitas around citrina, or what used to be called citrina, have proper published names. Maybe that's in the works, but to my knowledge, nothing has been published in the literature to make these names valid or accredited. I'm not sure what the proper
I'd
word
like to make one comment on the morphology that's pretty typical in this species. So a beginner might confuse the concepts of striate cap and innately fibrils embedded in the cap cuticle. These are innate fibrils. They are radially arranged, but they're just like part of the cuticle of the cap. They're like, and I don't think they're actual really fibrils. They just look that way. Striations are actually like grooves that are how do I want to say this?
They are the basis of the gills attached to the tissue of the cap, and the tissue of
Yeah,
the cap is very thin, so they're kind of shining through.
but you see actual like depressions in between ridges, sort of when
Right.
it's striations.
I was trying to describe this this week to someone, and the best I came up with was not only the gills but the absence of the gills. So you're seeing, if that makes any sense, you're seeing both of those depressions.
Why don't you just tell them it's like the folds in the umbrella? Like like
That
for
would be plicate.
like, well, I mean, like for like the way that striations show up, it's kind of like
Similar, but that's a more like extreme version of striate.
I think
It's called plicate.
strike, scratched versus streaky. This is more streaky. It's not like a sign of any scratch.
Yeah, there's no actual depressions radially arranged.
These fibrils is basically the way the pigment is arranged. It's it's an optical illusion. It's not even physical fibrils. It's like a sunburst pattern of color. The color is not uniform. You get these streaks, and they're just radially arranged.
Yeah, that's a good way of saying it. It's like an optical illusion.
Yeah,
really.
it's like
it's not
painted.
anything actually there except pigment.
They are painted essentially. That's what it is.
Yeah.
There's nothing physical, physically associated with the pattern. It's not like a raised groove that's streaking across the cap. It's just the variation of the pigment, intensity.
Also, about this species, the basal structure can be highly variable. Sometimes you don't see much down there at all because it might break apart.
Yeah, but it's it's good. It's good to keep in mind that the bulb, this is probably the classic look of it. How it should look like when the mushroom is in
I
good
agree.
condition.
This is what you usually see.
Yeah.
With short limbate, you can say, I guess.
And also on the smell, if it does smell like raw potato, awesome. But I've found, I think, what I would call solaniolens several times, and don't pick up on that odor very often.
Yeah, odors are not reliable. Some people don't smell things at all, and
Yeah, I have a bad sniffer also, but
yeah,
I don't.
And
my
the
schnozzle smells pretty
lavendula
good.
types also smell like raw potatoes, and those could possibly be confused with this, except the cap color is usually different.
Yeah, Amanita lavendula smells like potatoes as well. I'm not sure about brunescence.
Brunescence is also, I believe. And then there's these other provisional species, Corneli hybridium.
Yeah, I
Which
forget
Yeah,
is
about that one. Yeah,
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 I think there's.
and Amera citrina.
Americitrina. Yeah, those are the in the lavendula group.
Yes,
They look more like lavendula than. I mean, this species here is very distinct. You cannot really confuse it with anything else. Once you really zoom in onto those features here, the bulb, the small yellow ring, and the odor and the sunburst pattern on the cap, which is brown, you probably arrive to that ID pretty quickly. This thing is very distinct, at least in my eye. Lyle and I saw another example of the species when we went for a separate walk after the foray, and it looked pretty much like this one.
I can pop it up quick if
Yeah,
you
go ahead.
want.
I have to stop sharing
Yeah.
for a moment, and you can produce that photograph. There you go.
All right.
And I'm going to go over one more collection, which is nameless, so maybe somebody can point out a good idea, but that discussion is going to be very short. So
All right.
I'll
So
leave it here.
just quickly going through, here's that starburst disc.
yeah, and this
effect.
is what I'm talking about, where a novice might interpret this as marginally striate.
Right, these are not marginal striations. They
Yeah,
almost look like fibrils, but again, these are not true
hmm
fibrils. It's just a variation in the pigment.
yeah. That's an excellent example of what I was trying to explain. Thank
Yeah.
you.
Do you have more photographs of this?
Mm.
Yeah. Small ring tinged with
Yeah.
yellow. Maybe not as much in this one. A
A little bit of yellow.
little bit of yellow. Yeah, it's going to develop yellow tinges later on in life. Maybe. Yeah, and this, yeah, same bulb. You see that limbate quality to it. This one is a bit more mangled because I think the soil was more moist, and there's been probably some tissue damage because of that, just waterlogged. But pretty much the same kind of morphology of the basal vulva,
Yeah.
a bulb with some limbs, giving an impression of a secate vulva. Small size too. This one was identically sized.
Just a quick view of the gills. Yeah, a nice one.
Yeah,
Okay.
it's not a very common species, but once in a while you'll see these, maybe quite a few in a particular year. One more collection I wanted to share and maybe hopefully get a name from somebody. Let me go back to the list. Yeah, it's this one. Would be nice at least to get some genus name. They smelled pretty nicely, very fragrant, sweet odor. Very decurrent gills.
Yeah.
I don't think this is a live phylum. Nina said it was a live phylum. Live phylum doesn't have.
It was growing in the ground.
I don't know. Somebody brought this in, and I have no idea who it was or where it came from.
I
I
think
can.
I've shown
How
this
how deep
before.
are the gills? Are the gills very shallow?
I think the gills are shallow.
Yeah. Hohenbuehelia.
Hohenbuehelia. Mm-hmm.
What's that long? Um, Hohenbuehelia. Because they have that sort of shoehorn shape when you see them from the other perspective.
Like from this perspective.
Yeah. You see, they have kind of the shoehorn shape.
Yeah.
Yeah. So my guess would be Hohenbuehelia. What's that species name?
I can probably look this up.
Yeah, I'll get it for you in a second.
Not petaloid.
Petaloides, yeah,
Petaloides.
petaloides, yeah.
Hmm. Okay,
That's the
well,
one.
I saved this collection, and we'll probably submit this for sequencing when the time comes in due course. But I thought it was unusual, and nobody could even sort of come up with a genus for this thing, and
Yeah,
it had a very pleasant odor, a very pleasant odor.
I'll say to Dave's point, they are very shoehorn shaped because I thought maybe from this angle they don't look dissimilar from old Pleuteus, whatever the yellow one is, Citrino.
Oh,
Pleuteus.
Citrinopileatus.
Yeah, but
Yeah, these were more brown and yellow. I mean, Pleuteus is a very easy species to ID.
yeah, I agree. But I think Dave, you're onto something with Hohenbuehelia.
Yeah, now most of the Hohenbuehelia have really interesting cystidia on the gills. So if somebody wants to scope this, they might be able to find something that really solidifies that, at least that genus ID. And if it's Hohenbuehelia, it's almost certainly petaloides because the other ones are smaller.
Right. Look at the gill edges here when they transition to this type have more of a jagged pattern. Maybe it is just some kind of a defect, quote unquote, not necessarily a trait. But you look at the gills that in the caps they have a straight edge, and here they're more white. There they're a little bit more yellow. But I'm pretty sure that the spore print is going to be white for this thing,
There's
even though
no way
there's no more spore print. These are dried.
there's no way this is like a Lentinalis cochleatus.
Those have serrate gill edges.
Okay. All right.
All right, guys. Thank you for your patience. I'm done. This is all I want to talk about, and I hope you enjoyed this discussion and maybe learned something from it.
Yeah.
I certainly did. Repetition is the mother of learning, as they say.
Yes.
Yes.
The older we get, the more repetition we need.
Sometimes too much repetition. I'm guilty of that. I know.
All right. So Dave and Brandon both emailed things. So Dave emailed in first, so I'm going to begin with his. So let me get his up. And let's try to go through these fairly quickly, Dave, so we can get to Brandon's.
Okay, now you can hear me. All right, I'm ready to go.
All right. Oh, I've shared the wrong screen. All right, hang on. Share. Okay.
Okay, let's make this first one quick because I mean, if somebody thinks they might know this, they're probably going to be able to say so rather quickly. It's just this resupinate patch of bumpy fungal material on a dead branch of a living apple tree in my yard. I took a sample for DNA, but I have no idea what this is. And I was wondering if somebody might have a suggestion. If not, we can just run through the pictures really fast
Well,
and we don't have to dwell on it.
I mean, this is it's cool. The Hymenium just smooth, no
It's
pores.
just bumpy. I don't see any pores or anything.
Well,
I just see like a lot of bumps.
It's a little
Marioid.
bit wrinkled. Yeah, Morelia. Yeah.
Yeah, Mariliolite, but it's not doesn't have that kind of texture. This is really hard.
Yeah, they're almost wanting to shoot out into pores there.
Yeah, it seems like that. And this is actually pretty big too. Um, it's maybe 10 inches, 12 inches long. This and there's other smaller patches on the same branch.
And was the margin, I mean, maybe it's dried out, but was the margin membranous or was it kind of wispy?
It's
Yeah, and it
just
looks,
it's the whole thing is hard.
yeah, yeah,
It's
okay.
it's pretty rigid, and it's the whole thing's just plastered onto the tree branch. Yeah, there's
Maybe an old
not a
flat,
whole lot to say about this really. I just
On the decorticated part of the branch, it looks like, yeah.
yeah, yes, not on bark. Yeah,
maybe an old Flebia or something. It's my best
I
guess,
don't
but
think so
yeah,
because this is in my backyard. If
you
it
would
was
have
an old
seen,
Flibia, I would have seen it,
yeah,
like last year or something.
uh,
But
that's
that's an interesting guess.
that's about all I got on it. It's cool.
Eventually, I'm going to harvest some of this and see if I can play around with it.
Okay. Keep us updated.
Okay, so that's a close up. There's another photo that shows more or less. So this is Willis. It was found in an area where there is white pine, but no other kinds of pines. There were also hemlocks. So at first I thought, oh, it's a slam dunk. It's just a young Suillus weaverae. But there's another species that this one brings to mind, mainly because the enrolled cap margin seems to have maybe some, like a little flap, like maybe a little extension. Um, and that would be what it used to be, I think, subvelutipes, but now there's this new name, glandulostipes. So so now I'm stuck. I don't know which one this is, and I wish I would have sampled it, and I didn't because I thought, oh, it's just a weaverae, and it was at a club foray, and I already had so many things filling up my little compartments for samples. So I didn't even bring it home. I just photographed it and left it there.
Dave, we found something similar at Stokes, and I didn't want to put the name Wiveray on it, and it got a name, but it kind of looked similar, but it was older. But the cap color was, again, not typical of Wiveray. It had something else to it. I think the name for glandulostipes, the earlier name, or the younger name is Neoboletus.
I'm I'm sorry. Yes, Neoalbidipes. Right. Yes, that's
Yes.
right. I said something else with that was wrong.
Yeah,
Yeah,
you
it used to
said
be
salutations.
Neoalbidipes.
You
Right.
said salutations, and that's probably something
No,
different.
that's something else. Okay.
Yeah.
Yeah, I just got my names confused. Thank you. Yeah,
So yeah,
Neoalbidipes, which I have found. When you find it when it's young enough, you can. It's not that hard to ID because it has this
cottony
covering
margins.
over the hymenium that is not a partial veil or a universal veil. It's just a really long extension of the cap margin. And so that makes that species easy to identify when it's in that state. Now, maybe this smaller one had that, but I didn't check closely enough because we were in an area where I almost always find weaverae to begin with. It was right at the end of our foray. It was already over 3.5 hours walking up this trail. I was getting tired. So I didn't give this the right amount of attention, really. But I just thought, but I did get these photos and it looks pretty interesting.
Yeah, I would say this is not your typical Wolverine. It looks like it's something else. And
Yeah.
Neolabidipes or glandulostipes, what it's called now, glandulostipes, is a good possibility.
Okay, thanks for verifying my suspicions here. I wish I would have brought this home and sequenced it. Okay, so look at this thing. I'm
Tiny.
pretty sure this is a gilled bolete, believe it or not.
Let's see the gills.
Look at the gill. Look at the gill attachment. It's like adnext. Look at the
Yeah.
cap, though, and look at the spores. I got spores out of this. This one I did sample for sequencing.
This isn't bluing. This is like a moss or something.
That's not bluing. That's some moss stuck to
Okay.
the stipe. Yeah. Look at the next photo though. The spores.
Oh yeah, they're bolete spores for sure.
Holy spores!
Bolete spores, yeah.
Isn't that crazy? I'm pretty sure it's a gilled bolete.
Yeah, the question is what species?
Aren't there
Well,
like quite a few of them now? Haven't they decided that there's a number of those?
well, there's a few that are probably not named yet.
Yeah.
Yeah, it's still a small genus. It's probably 6 species altogether, give or take.
Yeah, I was going to
Not
say.
a very large number, but we still have to find out, what is Phylloporus leucomycelinus and what is rhododendron.
I think Leucomycelinus seems, I've submitted some of those like to NAMP and I think Fundus and maybe also
No,
there seems to be a lot of matching sequences that are called
there's more than one species of Phylloporus with a white mycelium.
Oh, okay. All right. Yeah, maybe in my area it's mainly just the one.
We're probably correct about our species concept for Leucomycelinus, but this has not been proven. It's still a conjecture at this point. Even though we could be right, eventually, after all. But we still have to prove this definitively.
Well, hopefully sequencing is successful on this thing because it's going to be interesting if we find out.
The ones I've seen tend to have more sort of decurrent looking gills.
Absolutely. That's what's an enigma about this thing.
Yeah, that's so interesting. Yeah, interesting. Yeah, it almost looks like a waxy cap or something.
Right.
Yeah, that's really neat. Oh, I can't wait to hear what it is.
And the spore print was pigmented. It wasn't a real thick print, so I didn't get a real good read on the color, but it was some sort of, light brownish, maybe a little bit of green mixed in, but I might just be projecting my expectation. But it was, it was pigmented.
I find the feel of the cap is like really telling with those two. It's cushiony,
velvety.
Yeah, and you can like push into it like squishy, like a bully.
Yeah.
Did it have that?
I didn't subject that sort of analysis to it.
So tiny. Wow. It would be hard to
do. It was
do.
Very small.
Yeah. They remember that this unusual and quite distinct species of Phylloporus that's probably undescribed. I think Steve Russell gave it a provisional code of Indiana 01. And I think your collection and my collection were the first ones. And I wanted to give it a name, but Steve Russell published it before I could come up with a proper epithet for a provisional name. And it's known as Phylloporus species Indiana 01. You should look it up and see if it looks similar. I think that one has gills that are tinged with red or they're very dark yellow.
Yeah, I think that one is one that I found in Oakwoods in a different location.
Yeah, you see, this one kind of gives me those vibes.
Yeah.
That could be Indiana.
I'll compare with that. Thanks.
Yeah, that one I think has a yellow mycelium at the bottom, not white.
Well, this picture is a little bit ambiguous because I had to like edit out some of the warmth, and I'm not sure if this base is white or maybe pale yellow. It looks like it was probably white, and it's just the camera's just adding a little bit of yellow that I
possibly. Yeah. So, well, hopefully sequencing is going to really tell us what it is because all of the sequences, all the ITS sequences in Phylloporus are well resolved. There's like no ambiguity, like, it could be this species, maybe the other one, just because we're like 5 bases away or something. They're all quite divergent.
All right. Cool.
Yeah, that was a good one, I thought.
Yeah.
So, Flavoconium, elongatum. Well, I thought maybe elongate them because of the white stalk. Now the cap is not very yellow though. But here's the problem. Look at the spores. If you can go through these pictures kind of quickly, you can stop on the next one because the next one shows at least the skirt, the ring and the gills. But where do you see the spores? And I took picture of spores from 3 different regions on the slide. So this is moving around the spore print. They're like subglobose, broadly ellipsoid. That doesn't fit the flavoconia concept. So I went on to Rod-Tulos's website, which is now being sponsored by Mushroom Observer, actually. And I found this variety, inquinata or whatever. It's there, and that one is supposedly has spores that are subglobose. So
Yeah,
a pretty interesting one, I would say.
yeah, it's a species group, and figuring this out is going to take some time. But I want to emphasize again that this species is a member of section Validae, and you can see all
Yes.
the features of section Validae in this particular species and specimen. The bright colors, the pronounced basal bulb. I mean, it's not like really screaming like a big bulb, but it is a bulb nonetheless.
Yes. Mm-hmm.
The persistent partial veil, and friable, something I forgot to mention, friable universal veil. In Validae, the veil is crumbly and flakes off pretty easily. And this is another feature of that section, important feature. But when you put all these facts together, section Validae is the only one that makes sense. And with enough experience, you'll be able to adhere Amanitas to section pretty quickly.
Yeah, the only one that's a little tricky is a species in section Amanita called Frostiana that can be mistaken for something that would be in the Flavoconia group.
Correct,
Except
but that one
Frostiana has a truly striate cap margin.
Correct.
And rolled sock basal vulva.
Exactly. The vulva, the structure, the shape of the vulva at the base of the stipe is very different.
Yes.
It's actually more related probably to the muscaroid group than anything else.
Yes, yeah, I think so. I asked Rod once if he thinks it should be in series Panthereniae, and he said yes, but he didn't say so in a really convincing sort of way. I mean, the morphology kind of matches, especially the basal structure.
Yeah, I should go and take a look at the ITS sequences of Frostiana and blast them and see where they fall in.
Yeah, see what else is in there.
Yeah.
Yes, yeah. Look at these spores, though. That's crazy. I mean, I thought I made a mistake or something, but no, I didn't bring home any other amanitas to look at spores with.
There's one spore here that is a very good orientation that actually tells you about its true shape. It's the one that sort of, if you draw a line from the tip of the arrow, you see 2 spores first, and then there's a second pair kind of slightly above.
Yeah.
Oh, above. No, above to the side.
Over here?
No, higher. No, not as high, no.
These two, no, yeah, the one you can see the
apiculus. Right, toward the right. Second, yeah, no, not the spare, the other one. To the right. I didn't say no, up and right. You're missing them. You keep no.
I only see these two. I see these two.
No, the ones.
These two.
No, so to the south.
Down to the left a little.
Southeast.
Southeast. These two?
No, not that far. You keep missing that pair.
This pair.
Not the spare, slightly to the right and above. Here, right? Yeah, don't you see them? That
no, pair. No, there's nothing there on my screen.
Well, you're circling on them.
Wow, it's nothing on my screen there.
Same. I cannot see anything there.
I mean, I see it perfectly.
Maybe you're on a different image.
You might see a different photo for some reason. Look at the other photos of the spores. Let's see if.
Oh yeah.
No, not this
one.
photograph. Not this photograph.
There we go. Probably
not this one. No.
Yeah.
No, not this one.
Wow.
The other one. This one.
Yeah, there's nothing, there's no spores in this area.
No, there's spores here. You are circling right. There are two spores, and one of them is in the proper orientation.
Huh.
Dave's arrow, his cursor
He
on that
points at
image.
these
Actually, it's
two.
mine. Never mind. Can you expand the photograph?
Yeah, it's not going to expand very much because it's cropped.
Well, I'm seeing the entire picture, and I'm seeing two spores that are further away from the arrow, slightly to the up, but are to the right.
Wow.
And one of
Yeah,
them is
I
in
don't
the
see
correct.
them either. Let me
No.
look at my photo.
Jeez.
Let me look at my photo that's not posted.
That's as much as I can blow this up, I guess. Doesn't
Oh,
the one
I
doing
wonder
it.
if I can. Yeah, I don't know. But I just want to point out that it's very important to look at the spores in the proper orientation. It has to be in the profile view, apiculus to the side. You're looking at, like, imagine a ship floating in the ocean. You're looking from the shore when the ship is in the profile, not from above, not from below, but right, looking at the broadside. So, and that's the proper orientation of the spore. You have to be looking from the side in the profile view.
And what this one looks like, kind of lemon shaped vaguely down here, slightly pointy on either end.
It's funny that where you're circling right now, what I'm seeing on the screen is just empty space.
Oh my gosh, that's so weird.
Weird.
Yeah, because there's a nice, almost, you know, squat lemon shaped one right there.
Yeah.
Well, I'm fit to window at 77%. If I go to the original image, how about trying to circle again while you're circling?
Here.
Nope, you're circling an empty space in
Yeah.
Dave's
Oh,
image.
that's really weird because she's circling a spore.
Yeah, right
On
here
my
is a
screen.
spore. A nice.
And,
Okay,
and
so,
where you're saying there's two spores, that there's nothing there on my
all
screen.
right. So you see the arrow of the reticle,
Yeah.
Yes.
and then there are going to be two spores directly to the right of that,
Yeah.
Yes.
They're
and there's going to be
touching.
another pair slightly above and to the right of that.
Yeah,
Right.
that's those two that Lila's.
Okay, but Lala's actually, her cursor is somewhere else on my screen, interestingly.
Oh,
But the
that's
bottom
weird.
spore, the bottom spore in that pair, where you can clearly see an apiculus, is in the proper orientation,
Okay.
Yeah,
Yeah.
and it's subglobose, almost like
it's subglobose.
borderline broadly ellipsoid.
Yeah, but this is way different than what you're supposed to see for flavoconium.
I understand, but I was just
Yeah.
trying to point out, when you look at the
Yeah.
spores and judge the size and the shape, you have to really look at the correct orientation, especially for spores that are not round or that are ellipsoid. Ellipsoid spores are very difficult to visualize properly.
All right. Well, you had one more, Dave.
Okay. So this is a species I've never attempted to ID before. It's a clavulina. What's the species name?
Ornatopes.
Ornatopes. Yeah. So this matches pretty well with what is pictured in Quebec under that species name. And this was also kind of interesting where it was growing. It was growing in kind of a wet, mossy area. Pretty wet actually. It was actually a little, very small stream flowing over some rocks, and the rocks were very mossy, and there may have been, or it may have been just soil under the moss. I'm not sure, but anyway, these look like a pretty good match for this thing that ornatopes that I found on Quebec. So I tried to take a sample of this and I dried it out. So it's another one that maybe we'll find out what it is for real. But that's my proposal as of right now.
Lila, can you go to the picture with the coin, please? And can you please put your cursor in the middle of the coin?
There it is. Right on Lincoln. Although you can't see him in there.
Well, the funny thing is that I don't see your cursor at all, and it's not on the coin.
Hmm.
Wow. But you see the coin.
I see the coin, and right now Lila's cursor is below the coin, closer to the end, but outside of the coin.
Oh, that's weird.
That's
Yeah,
how.
but that explains why we couldn't find those two spores you wanted to
Yeah,
see.
I know. But my question is, when I show my say observations to you, and I'm circling on a feature of a mushroom as I see it on my screen, do you see the same thing or not? That would actually worries me. But I don't understand how the presentation here, on my screen is different.
Yeah, I don't. I'll have to think about that. I don't know.
Right now, your cursor is barely touching the bottom edge of the coin.
Right at the top of the steps in the center part of the, right where Lincoln
Are
should be
you in
seated.
full screen
Yeah, I
view?
was going to say, Igor, didn't you say you're at 77%? If you make
Well,
it,
I'm looking at the view options, and right now I'm fit at fit to window at 77%. If I go to say 100% original size, I cannot even see half of the image on my screen.
Oh.
And when I go to 200 or 150%, I only see the top part of the photograph. The bottom part where the coin is completely gone.
Huh.
So I have a Chromebook, and it's like maybe like what 12 inches across. So I'm always at fit to window because that's probably the best presentation of the for the computer.
and
I mean, I can go to
what is
50%.
it? It's Clavaria coralloides, or
Usually that's bigger and it grows like in needles under conifers. But yeah, it could be that.
You do any chemicals?
I didn't bother to do anything with these because I just wanted to preserve them because it's only a small sample. There was perhaps a little bit more growing in this spot.
What
Um,
color were these in real life?
Pretty much like this, kind of a beige gray sort of color.
Yeah. Okay.
So
Yeah, I
Jim,
didn't harvest them. We were on our way out down the trail, and the spot was actually a little tricky. It was slippery. It was like this little tiny stream of water was going under a little footbridge. So one of the members who was with me harvested these for me, and there was not a real big fruiting, but there were a few more of them. But they were all small like this.
there is a button on the screen at the upper right corner says view. So I clicked on that and I switched from standard to side by side gallery format. So right now, Lila's cursor again is outside of the coin, just below it. So
Uh,
even though the image got bigger and the screen got centered, there's no change in the position of the cursor as far as I can tell.
So Dave, this is not cristata.
That's the same thing as correlades, right? And
Yeah.
it could be. It could
Okay.
be small ones of those. Um, but those, like I said, those usually, occur in larger clusters on drier ground.
Okay. Um, and then with your Amanita, it, did you consider Amanita erythrocephala?
That's European.
I think it's here.
No, I didn't.
I
Let
think
me.
it's number.
What's the first letter of that?
I put it in the chat.
Oh, okay. Yeah, let me see.
People say that, but there's no genetic foundation for that claim.
Rhythocephala. Yeah, well, I'm still going to look. I'm going to look on um, the Amanita website and just see what
See what this
what is said about that species.
Don't forget that European and American Amanitas, there's like no overlap in species.
Right, but there might be a North American counterpart to that, and that might be, this thing that I proposed. But at least, it would be interesting to look and see. Okay, so Amanita.
Great. It's 9. I can't stay on too much longer. I want to get to Brandon's stuff.
Yeah, go to Brandon's.
All right. You've got 4, Brandon. Let's see. Why is this not loading? There it is. A rust.
Yeah, I mean, it's a rust. It's a cool one.
It is.
It's on a plant that I was unfamiliar with. It seems like the alternate host is even more unusual. But yeah, the plant is
Looks like some kind of milkweed, maybe.
yeah, Decadon, minia. Oh no, that's the species of the rust. The rust is Puccinia, miniato-ovaceus, I think it is.
Yeah.
Zoomed in, it looked like little
Decadon.
cups.
Yeah, verticulatus, verticulatus.
Okay. I have to look that one up. That's a new one to me too.
Yeah, it honestly, I saw the plant first, just because it grows in swampy areas, and this was found between 2 cranberry bogs. And I mean, the actual fruiting body of the rust are the little cups, um, and it deforms this,
The
you know,
whole leaf.
the whole leaf and the flowers that would eventually come out between each one, maybe.
Mhm.
neat. That's cool.
Hmm.
Yeah.
I think the flowers on this, right, would mostly be at the top.
Mostly. So, you're right.
Those are probably more branches in the
Yeah,
leaf is that's probably a more of a branching spot. But if it's one of these like weird little swampy milkweeds or whatever, the flowers would be mostly up at the top.
it's a different thing than a milkweed. It's a uh, there's a common name on here, and I don't know my botany all that well. Jason helped me identify the thing. Uh, Shoot, I have it open in another window.
All right. Let's see.
Loose strafe, loose
Is it a
swamp.
loose stripe?
Yeah, swamp loose strife,
Okay.
or water willow, as it's sometimes called. But I do think there are flowers that come out of that kind of midsection. But the alternate host, if we want to go to the last couple photos,
Okay. I'll go back to it. Sorry. I jumped over here.
yeah, my bad. I'm in the wrong window too.
All right. All right. The last couple of photos.
Yeah, so this is where it was found, just the kind of general habitat.
Mhm.
And the alternate host is a sedge. And I think maybe in the last photo in particular, you can see some sedges kind of in the beginning of the water line.
Yeah, we're along the water edge here, and these look like sedges here.
But I didn't know that until after I kind of looked into it a little bit. And I'll put a link in the chat for anyone that's still here. There's a really cool article about this particular rust fungus.
Nice.
So,
Thank you.
yeah, if anyone ever is looking at any research and sees the Cornell mushroom blog, click on that. They're good articles, little write-ups, historical facts about how people identified rusts. The guy that named this thing, the mycologist Arthur, last name, uh, he said to identify this one in particular took 2,100 packets of spores. So, because you got to find out the alternate host for it to even fruit in the lab. You can't just grow it on a petri dish, you know?
Mm. That's cool. So what is the alternate host?
It said, she said, right?
Oh, okay. Mm-hmm.
Yeah, I think the genus is Carex, and it's a
Yeah.
specific species.
Of sect. Yeah.
Carex, Carex, I think it is.
Yeah, that's the genus for sedges. Yep.
Okay. Wow.
Cool.
Good
But
find.
yeah,
All right, yeah.
yeah, we can move on.
All right. I gotta get back to email. 10 feet up, standing dead pitch pine.
Yes, this was cool.
Yeah. Wow.
So growing, I had to take that plate off. I tried to extract it. I thought maybe it's just plate deep, right? And it's just growing up there. Nope, coming out of something alive. So I couldn't get the base. You know, this tree's not 100% dead. It's not doing great, but
That's really cool.
you could tell it was a Pleuteus from the ground. But I poked it with a long stick to get it down.
Pleuteus and a conifer?
That's what on a standing dead that high up conifer. I thought that was really weird. Pitch pine.
Got to be a new species.
Oh, what the heck is the name of this one? It's a white one. Pleuteus.
It's pale. I wouldn't call it white.
Well, the time I found that it was white, but I think it was on hemlock. So I think I found Pleuteus on hemlock. And there was one that was I sent to Fredo Yesto, and he identified it. Look at that.
The picture right there.
Wow.
That tree's not doing too well.
Well, are
No, it's
those
not.
like holes from a
Yes.
woodpecker or something?
Yeah. Or some sort of boring bug, but I would assume on a pine it's a woodpecker.
Kaput.
Wow.
That's
Wow.
that tree is toast.
Yeah, the next photo gives you a little bit even better scale, I think.
So it was growing out of the a woodpecker hole, huh?
I don't even think it could have been. I mean, that would be the opportunistic way to do it.
That's what it looked like, you know. I mean, this thing is riddled with holes.
Mhm.
Yeah.
But look at that! Look at that big polypore. That's even 20 feet up.
Yeah.
Yeah, I hope you didn't climb up that trunk.
I did not. No, I used a big stick to poke the Pluteus down. I couldn't reach the polypore. But again, I mean, look at how tall that thing is. It's kaput, like you said, Igor. But it's still holding strong,
Yeah.
which is another. I don't normally find Pluteus on standing dead pitch pine.
No, I mean, it's almost like that it was like introduced that high, you know, with a bird or something, maybe.
Mm.
You know, it's
Stuffing nuts in the holes or something.
I mean, it's like, you know, you'll find Pluteus on the ground or growing out of rotten wood, but you'll never find Pluteus growing out of a tree trunk, you know, like 10 feet above the ground or something, right?
Right. And I was thinking like, oh, maybe it's a situation like we talk about Tylopolis velius kind of crawling up and growing on a stump or something like that. But that stump is always pretty well, you know, uh,
Decayed.
disintegrated,
Yeah.
decayed.
Yeah,
Yeah.
the mycelium just travels from the ground up, through the woody, substrate when it's like that rotten. So it gives an impression of a sapro, but actually, I'm pretty sure that Tilapus felius is a mycorrhizal bolete.
Right.
But
And
yeah,
there are Pleuteus, and I did a little bit of research. I didn't do any micro. I saved the thing. I dried it. Um,
yeah, it's a good sequencing candidate. Very unusual to be growing on a conifer that high,
Yeah.
and
And a great specimen too. Jeez.
yeah. So cool. I thought that was at least interesting. It takes a very interesting Pleuteus to get me excited. So,
were there any
Nice.
other dead conifers nearby?
oh yeah, yeah. But no other Pleuteus that I found. And I looked around. I mean, obviously on the standing dead ones. This area was burned a couple years ago, so
Uh-huh.
I often find interesting, uh, specimens in that area.
Yeah, this looks like
It's a Russula. Maybe can you zoom in any further on any of that?
yeah,
Hmm.
it's one of the stinky ones. So maybe this is
Yes,
something like Rusula mutabilis or something like that.
that's what I wanted to call it. Although there were a couple things that kind of led me away from that. Um,
Not dark enough. Did you drop any KOH on the cap?
I didn't, but I should do that. Uh,
Yeah,
the color of the
definitely
study,
sounds.
it definitely stank.
Yeah,
And
it's that benzaldehyde, whatever, marashismo cherry odor, whatever they call it.
this marginate gill, if you can zoom in a little bit, maybe just click.
Oh,
That was pretty interesting. I mean,
mhm.
there's a couple of these fetid russellas that present reddish brown on the gill, and you can definitely see that the red kind of staining. Um, but that marginate thing, um, I don't know. This is there's a couple of these Phoetis, Mutabilis. There's a couple cryptic species.
Right.
Yeah.
It's definitely not ventricosipes. It's too early for that
That,
one.
I the stipe is what made me think ventricosipes, and this was not sturdy enough to be ventricosipes, but um, the marginate gills that you can even see in this photo on the sectioned one.
Yeah,
Um,
yeah. It's not some kind of a fake feature. This is quite real.
yes, yes, but the stipe is almost punctate and has reddish, yellowish things going on, which I was on Michael Quebec just kind of briefly looking at some of these fetid ones. Um, and it seems like there's more and more popping up. And I don't think this is ventricosipes.
Yeah, it doesn't look like it. I think we find ventricosipes in the fall, not in summer in the Pine Barrens. But again, my experience doesn't mean it's just
Well, it's also much bigger. If you look at the sectioned one, look at how thin the flesh is at the apex. And I understand that these are, uh, well, I mean, they're mature enough to have marginate gills. Look at how thin that flesh is, though. Normally, ventricosipes is a big, sturdy thing, which makes me think this is probably mutabilis or, one variation of mutabilis, something that might have a coded name related to mutabilis.
Yeah, I mean, when I see Mutabilis in the Pine Barrens, it's pretty unmistakable. I don't think I confuse it with any of the other, fetid rooseveltes from that section or subsection or whatever. Um, uh, the thing is that in the Pine Barrens, Ventricosipes is the only one that we find. We don't find any other ones, to my knowledge, or I can just recall finding, say, Mutabilis in the Pine Barrens or Russo Grata or all the other ones that have European names. Voraceraceae. Yeah, Ventricosipes is the only one that really stands out in the Pine Barrens, and this one doesn't give me the Ventricosipes vibe.
Yeah.
But they're certainly related, and that marginate guild thing, I cannot replace that.
Right. So
So
again,
Fragrantissima, that's another
Fragrantissima,
name.
that was the other
yes.
European name. Yeah. Mm-hmm.
By
And
the
then
way, I
it
put
is
something in the chat for you, Brandon. Your there's a Pleuteus that grows on conifer wood. Tomentosulus. I put a link in the chat.
yeah, and there's a few, and in my bit of research, they're all like well decayed logs, though, right? Does it mention that for that species?
No, but when I found Tomentosulus that was ID'd by Alfredo Yesto, he sequenced it. I believe it was on a standing tree, probably a hemlock.
Cool. Yeah, I'll definitely look at it. Oh, Amanita. Calling it Bronicensis for right now. It does have a rooting base. Got cut off. And no bulb, but
Can we take a look at the cap again? I don't think this is section Rhonacensis.
Yeah.
I don't think it is section Rhonacensis. I think this is Amanita submaculata.
Submaculata. Okay. Oh yeah.
Look at the cap and the vulva on it. Also, in submaculata, the stipe does stain red when you touch it.
Yep. And if you go a couple more photos, so this was maybe an hour later after I handled it, and you can already see some of that reddening. This next photo probably shows it even better.
Yep.
Would submaculata have these small patches of UV material on the cap? Doesn't that one usually just have like one or two big patches, if anything? And the bulb is usually smooth.
I mean, the only other Validae that is a Rhonacensis wannabe I can think of is Canescens.
Oh. That looks a little different than this also, but I think this looks more like canescens than submaculata.
In Canescens, there's no reddening of the stipe, but there are these orange fibrils that rod buses in handling. I mean, this would be kind of extreme. But one thing for sure, I don't think this is a quote unquote lepidella in a broad sense.
I wouldn't rule it out. Lepidella, they're strange. There's all kind of strange morphology in that group.
Yeah, well, it gives me a Validae vibe, and it's a Validae that is early divergent.
Why don't most of the lepidellas have like? I might be thinking of Amidella, but do they have like long spores, like bacilliform spores?
Well, bacilliform spores, I think, involved with ecology, and they are quite a common occurrence in quite a few species of pine barrens and Amanitas, especially from sectional lepidella. But
Yeah, that's what I'm thinking. It's
Yeah.
you know, if look at the spores, because I don't think you have Validae has any species that have spores like that.
Yeah, Validae spores are mostly ellipsoid. They're not.
Ellipsoid or
They're not
broadly
elongate, or
ellipsoid
certainly
or
not bacilliform. Never.
sub globose, some of them.
Yeah.
Oh, was that one picture showing an intact partial veil underneath? Because that did look more like Validae. Yeah, that one. That photo.
Yeah, to me, again, the properties of the cap, the bold cap, and the disposition and the form of the veil patches, velar patches, is more typical of Validae from those early divergent
The cap
species like
doesn't look
Canescens.
bald to me, though. It looks like it has all these little warts all over it.
Yeah, the cap is not bald, and they're powdery.
No, I mean,
Powdery warts.
I see the warts, but the rest of the cap where the warts are absent, that's glabrous. That's very shiny and bold.
Yeah. Oh, yeah. That's true.
Wow.
I
was also basing this a little bit on an appendiculate kind of thing going on the cap margin. Maybe that wasn't photographed all that well.
Yeah, but it looks like that stuff might be PV material.
Cool. Yeah.
No, appendiculations are always partial veil material, but sometimes you have universal veil patches that are like on the edge of the cap, giving an impression of appendiculations. But in fact, there are remnants of the universal veil clinging to the edges of the margin of the cap.
I thought that the appendiculate material in the lepidellas was UV material.
No, it's PV.
It is? Oh.
Yes. I think appendiculate material is always partial veil. No matter the section. It's the same thing. So check out if you go to
Validae. What did you say? Not multisquamosa.
Submaculata and canescens.
Look up these two species. Also look them up on Mushroom Observer. There are some good IDs.
Also, canescens is big, right? Isn't that
canescens is big? Yeah, canescens is big.
Okay. All right, cool.
Yeah, so is submaculata. That's also big.
Yeah, submaculata can grow tall, but it's not as clunky as canescens.
Yeah,
it's more graceful.
Mm-hmm.
Both are found in the pine barrens. Been confirmed by DNA. Definitely submaculata and canescens. I don't think I've sequenced canescens. Maybe I have. But that species is more sort of has a better understood or more stable morphological envelope.
All right. Well, cool. Thank you, Lila. You've hung around. Way to be a trooper. Appreciate that.
Yeah, we got started a little late. So, all right. Thanks, everyone. Hope you enjoyed the session. See you here next Tuesday.
Thank you, Lila.
Thank you.
Thanks, Lila.
Yeah.
Nice session.
Yeah, that was fun. It's good.
Do you know how to end the recording and keep it open? I guess you got to stay logged on, huh?
Let's see. I got to stop the recording.
You have to stop the recording, yeah, and then you can.